can we have a way to induce more broad immune protection than the current vaccine? But the current vaccine is working very well with the original virus strain, but not good enough for the variants like Alpha, Beta, Delta. The findings are very exciting. Say, yes, we can do it, but only happens in the patients who survived the SARS-1 virus infection 17 years ago. So now, if you immunize with the SARS-2, then you produce what we call this broad protective immune response. And we measure one of the arm of the immunity called the neutralizing antibodies. Yeah.
So after all this time, people who had SARS-1 maybe up to 17 years ago, they still retain a degree of antibodies? Because I know there's been concern about how long the antibodies last in people with the SARS-CoV-2.
So we know already that, so we published last year about maybe, March, April to say the SARS-1 patients 17 years later, yes, in their body, they still have the circulating. What we mean by circulating is in their blood system. They still have these neutralizing antibodies, but only neutralize the SARS-1 virus, and they could not neutralize SARS-2.
Right.
Okay.
Right.
Yeah. But now, after vaccination, they can neutralize both. Yeah.
Because just to explain, a lot of people maybe don't realize that the COVID-19 is basically the disease one gets from SARS-CoV-2, and it is a SARS virus. It is from the same coronavirus family.
Yeah. So I used to, describe them as either sisters or close cousins. Basically, in scientific classification, we classify them as the same species. Okay, at the virus level, and then you have a different strain or different clade. We call different clade. Basically, you have the clade 1, SARS-1-like virus. So one of them jumped from bats, we believe, and caused disease in human. And then we have many, many SARS-2-like viruses exist in bats and pangolins, and one of them jumped and caused COVID-19. Yeah.
What processes are you looking at? I know you've been looking at a molecule that it's taken in the process of infection, and you're trying to stop that. So can you just explain what you're focusing on?
Yeah. So this discovery have two parts, actually, the publication. The first part is technology. I always say, as a scientist, you can have a brilliant idea, but you need to have the tools. So we actually invented our own tools to measure the neutralizing antibody with subtle difference. So now we can measure neutralizing antibody against the original strain, Alpha, Beta, SARS-1, and the pangolin virus, bat virus. All of these are in the scientific name called them sarbecovirus, basically a subgroup of viruses. So in this, we invented that technology. We can measure neutral antibody against 10 different sarbecovirus in one go, and all we needed is a drop of your blood, few microliters. So that's the part, really kind of a clever, if you can use that word, a clever kind of design to have the tool. With that tool, now we can interrogate antibodies in different sort of groups. So we found that if you are just infected with SARS-1 or SARS-2, or you're just vaccinated with SARS-2, you don't induce this broad protective neutral antibody. Only the SARS-1, now 17 years later, vaccinated, you have this quality of antibodies, which can neutralize all 10 different viruses. And when we say neutralization, it's a broad term. In this study, we're specifically referring to the ability of antibody to block the virus bonds to a molecule on our cell surface called a receptor. So that binding is essential for virus to enter. Okay, so if you block that, basically you block infection, and then you don't worry about disease, right?
Why do you think it is that at the moment we're concerned about different variants of SARS-CoV-2, meaning that current vaccines are not as effective? How does this therefore give you a much more broader protection?
Yeah, so for your audience, this is a very kind of a sophisticated science, right? Immunology. So our body evolved very, very sophisticated. So we have many different ways of fighting infection. And one of the arms is producing antibodies. And then the most functional antibody is the neutral antibody I just described. The antibody molecules are like the key. We have millions, if not billions, of these molecules in our gene. You're born with that. But they only produce the keys absolutely necessary to open the locker and to fight the virus. So it's like the smart system, so that they only produce the antibodies very specific to virus. For example, the original virus. And when you have the alpha infection, you produce a different set of keys. And then if you have a delta infection, you produce another different set of keys. But because the alpha and the delta are so close related, most of the keys will be sharing. But the most essential key, which really unlock this, the receptor and the virus binding, are very specific. So it's good if you only have one virus, because you don't waste the energy to produce the keys which are not useful. But when you have a variant appear, then the very specific keys are now you cannot open the doors to the other variant. That's why you afford some protection, but not 100%. Mm-hmm. So what we want is that from this study to say, instead of producing very specific keys, we still produce a very limited subset of keys, but these are the master keys. Mm. They can open doors to alpha, beta, and the SARS-1 and the virus yet to emerge, like the SARS-3, SARS-4, So this is, in layman's term, I try to explain that way because our body's immune system is very, very complicated. So what we try to teach our immune system is to say, yes, still produce a subset, but can you produce the broad spectrum master keys instead of the specific keys?
So how excited are you at these findings?
Very excited. You can tell from my body language. As a scientist, I have been in this game for almost, 35 years now, right? And you have this, what we call eureka moment, really, really top scientific discovery, and something that is not easily predictable. So you have a gut feeling, hypothesis, it may work, but the chance of success is low, but the impact is high. So in my life, scientific career, it only happened twice. The first time was 2005, when I also doing this kind of, immunity studies, and then discovered that bats carries antibodies to the SARS-1 virus. So then led to the discovery to say bats are the reservoir of SARS-1. So that was the first eureka movement. And then, that's 2005. So almost, 20 years later, 18 years later, I had the second eureka moment is I discovered that, wow, my God, the antibodies, neutral antibodies, only in that group of SARS patients now vaccinated with the SARS-2 vaccine give this ideal immunity, this broad and highly potent. Yeah.
So what's the next step in your research to explore this further?
So in terms of translation or, real impact on the ground, 3 levels. Number one, everybody is really dying for is a new generation vaccine. I have to emphasize this vaccine will be a boosting because we are trying to encourage your body to produce this cross-broad neutral antibody. And there's no better time than now because in the human, mankind's history, I would say, it's the first time we want to vaccinate the whole population with one vaccine against the SARS-CoV-2. So basically, by this time next year, majority of the world population, not only UK or Singapore, the world population will be primed, already receive vaccine against SARS-CoV-2. So if we can produce a new generation of boosting vaccine, which can induce that broad immunity, then you don't worry about variants anymore. Not only the variants now, the variants of tomorrow, because if you look at the genetic code, SARS-1 and SARS-2 in that key part, the receptor binding domain, they differ by 27%. Whereas alpha and beta differs by maybe 1% or less. So if you can protect the virus that 27% different, I can almost be certain that it will protect a variant. Another variant is well known 2% different. So that's the first layer. Some people call it a dream vaccine. I call it a next generation vaccine or third generation vaccine. The second layer is that By the same token, we have these monoclonal antibodies for therapy, right? We call them therapeutic monoclonal antibody. And President Trump was the most high-profile person who used it, okay? These are the antibodies derived to fight SARS-CoV-2. And that's just like the vaccine. We already have seen some of the original monoclonal antibodies are not as effective against the variant and maybe less effective against future variant or future, a SARS-like outbreak. So using the same principle, we discovered that from this cohort, we can clone and produce high potency and very broad neutral antibody for therapy. So that's the second impact sort of we are working on. The third one is the technology. As I said, this is a very sophisticated, we call it a very, the scientific name is multiplex surrogate virus neutralization test. Basically, it means that we can do this neutralization test without live virus. So it's a surrogate, and we can do 10 in this paper. Now we can do 20 in one go. What that means is that we can do risk assessment for the virus circulating in the animal and to see any of them jump to human. So for the same reason, if we can archive series, from, Wuhan or other places in the world, we can actually go back and help this tough question of how this virus originated and how they transmit early from animal to human. So this is the same tool that used in this particular study can be used to study virus transmission and the origin virus. So 3 different levels: vaccine, therapeutic monoclonal antibodies, and tracing the origin and the early transmission events, and the future risk assessment of similar viruses.
What is the likely timescale to progress this work to the time where we might be able to produce a vaccine?
That's the most difficult question for a scientist. I always say, a scientist, my responsibility is to make the discovery and point the direction. Then, of course, I'm working with partners, and we try to convince them, first of all, this is a good idea. Then the commercial partners will really, run with it, right? Because I'm not experienced, and also I'm not good at that. So the most, optimistic estimation, everybody says mRNA vaccines only take 6 weeks to produce, So we have the big companies doing that, and they're doing this for the beta and the delta. So if I can convince them to say now using this, SARS-1-like sequence, will maybe a better booster vaccine, and if they agree, and then they will take on this on board. If they, use that as a high priority, then within few months, we may have a candidate for clinical trial, right? You understand, if you have a vaccine, it's serious business. You have to do safety, you have to do efficacy before you can, register for a product. So I always say, the most optimistic, estimation is if we have some molecule for trial by end of this year, that would be pretty good. Yeah. When you think about it, right, I have to emphasize this is not a vaccine to replace the current vaccine. This is a vaccine to boost, on the basis of current vaccine. So consider that in our world now, still a lot of people have not even received the first vaccine yet. So that's what I'll say. On one hand, there's urgency. On the other hand, we still have time.
I know in the last few days, the Singapore government have said, this is something we're going to have to live with. We cannot eradicate it. And other world leaders and health agencies are recognizing that. So this is a long-term process and will become part of our armory going forward, potentially, to protect the human population against these types of coronaviruses.
Yeah, so this is live with the virus. I think it can be misinterpreted to say, you don't worry about infection and death. No, I think, when you balance everything, you cannot do the zero tolerance, like a certain country and certain cities can do in the beginning of the pandemic, And so eventually, I think that what you want to do is to reduce the risk of severe infection deaths as much as possible, but you live with the virus as a society. No. So some people thinking live virus as an individual. If we live virus as an individual, we fail, right? Live with the virus as society and control the severe disease and deaths to the minimum. And so people always refer to is influenza virus. We have seasonal flu, and this is the number one transmissible virus and kills more people than any other virus, and yet we have survived hundreds of years, with different kind of countermeasures. So that's the kind of context is for the society to live with it. But like what I'm doing right now, hopefully will make that live with virus much more comfortable, rather than at an individual level, you still worry about infection, if not today, next day or next month, so we can live as close to normal life as possible.